Hello and welcome dear students. Today we are going to discuss about the topic genotypic and allelic frequencies. The main objectives of today's lecture are to know the basics of genotypic and allelic frequencies, to understand the measurement of gene frequency, to discuss the factors affecting gene frequency, to understand the similarities between gene frequency and genotype frequency, to know the differences between genotype frequency and allele frequency, to understand the genetic inheritance and evolution, and finally to discuss about the allele frequency and evolution. Dear students, let's first start with the introduction. At present, population genetics has become a widely studied field by geneticists due to the popular trend of emerging species. Thus, population genetics can be measured through microevolution, where the evolution of a small population is analyzed in terms of its allele frequency or gene frequency, genotypic frequency and phenotypic frequency. These calculations are done in order to determine the similarity of a population and to develop evolutionary relationships amongst different species in a population over a period of time. The frequency determines the number of times a particular gene, genotype or phenotype is repeated in a particular population. The key difference between gene frequency and genotype frequency lies in the particular factor in which the frequency is determined. Genotype frequency and allele frequency are two parameters used in this. This equation calculates the genetic variation of a population at equilibrium. Study of the frequency of genes and genotypes in a Mendelian population is known as population genetics. In other words, it is a branch of genetics which deals with the frequency of genes and genotypes in Mendelian populations. Before dealing with population genetics, it is essential to define Mendelian population gene frequency and genotype frequency, that are random mating and equal survival of all genotypes. In case of random mating, each individual of one sex has equal chance of mating with every individual of opposite sex. In other words, there is no restriction on mating of one individual with other individuals. Such intermating populations are known as panmictic populations. Random mating, individuals belong to the same species and same gene pool. The gene pool refers to the sum total of all the genes in a Mendelian population. The allele frequency or gene frequency is the rate at which a specific allele appears within a population. In population genetics, the term evolution is defined as a change in the frequency of an allele in a population. Frequencies range from 0, present in no individuals, to 1, present in all individuals. The gene pool is the sum of all the alleles at all genes in a population. Using the ABO blood group system as an example, the frequency of one of the alleles, for example IA, is the number of copies of that allele divided by all the copies of the ABO gene in the population, that is, all the alleles. Allele frequencies can be expressed as a decimal or as a percent and always add up to 1 or 100% of the total population. For example, in a sample population of humans, the frequency of the IA allele might be 0.26, which would mean that 26% of the chromosomes in that population carry the IA allele. If we also know that the frequency of the second allele, that is IB allele, in this population is 0.14, then the frequency of the first, that is I allele, is 0.6, which we obtain by subtracting all the known allele frequencies from 1. Thus 1 minus 0.26 minus 0.14 is equal to 0.6. A change in any of these allele frequencies over time would constitute evolution in the population. Dear students, let's discuss about the gene allele frequency. Gene frequency can be defined as proportions of different alleles of a gene in a population, and in a particular generation these frequencies depend on their frequencies in the previous generation and also on the proportions of various genotypes in total population. In any population, if a character is controlled by two alleles, then the frequency of these alleles or genes can be calculated very easily by phenotypic observations of that character under homozygous and heterozygous conditions. The frequency of an allele in a population is the number of occurrence of that allele divided by the total number of alleles of that gene locus. The gene is the unit of heredity which is transferred from parent to the offspring generation. The information which governs the characteristics of the offspring is stored in these genes. Every gene exists in alternate pairs, and an allele is one alternate form of the gene. We can also say that alleles are the alternative forms of the gene. So gene frequency, which more or less refers to the allele frequency, is the measurement where the number of repeats of the same allele is measured over a certain period of time. Thus, gene frequency refers to how frequently an allele of a gene appears in a population. The allele frequency is the frequency of the two forms of a particular allele in a population. They are dominant and recessive alleles represented by capital and small letters. Each allele frequency can be calculated by dividing the number of individuals with the allele formed by the total number of individuals in the population. Allele frequency is just one aspect of population genetics. An example of population genetics can be seen when assessing the probability of which eye color, that is gene, from each parent, that is two alleles, will be seen in their children or population. Allele frequency is how often an allele develops in a population. When assessing the probability of eye color, parent 1 having blue eyes and parent 2 having brown eyes, allele frequency would determine how often blue and brown eyes would present in the population, that is the children. Alleles always are represented in pairs. One allele is passed from each parent for every gene type. Although the genotype or genetic makeup of an organism, that is DNA, may show a variation in different alleles, the phenotype of that organism or outward appearance will possibly favor only one allele. The ability of an allele to be expressed depends on whether they are dominant or recessive. Dear students, let's talk about the measurement of gene frequency. In a diploid species, a population having n individuals has 2n alleles for each gene locus. If there are two alleles, A and small a, of a particular gene, in this population the number of capital A alleles is twice the number of capital A homozygotes plus the number of capital A small a heterozygotes, as each homozygote has two capital A alleles and each heterozygote has one capital A allele. So the frequency of capital A allele is the number of capital A alleles divided by the total number of alleles, that is 2n. Dear students, let's now talk about the estimation of gene frequencies in a population consists of three important steps. Number one is sampling. First, a random sample of individual is drawn from the random mating population under study. The size of sample should be adequate to represent all the individuals of a population under study. Number 2 is the classification. After sampling, the individuals are grouped into different classes for a gene and their number is counted. And the third one is calculation of gene frequency. As an example, let's determine the percentage of each allele group. If A, that is capital A, is equal to 60%, then small p is equal to 0.6. And if small a is equal to 40%, then the q is equal to 0.4. So capital A frequency is 0.6 multiplied by 0.6 is equal to 0.36. While as in heterozygous condition, that is capital A small a, frequency is 0.6 multiplied by 0.4 is equal to 0.24. For homozygous allele small a, frequency is 0.4 multiplied by 0.4 is equal to 0.16. So out of 1000 offsprings, it can be expected that there will be 360 capital A individuals which are homozygous dominant. There will be 480 individuals of heterozygous type which will be having capital A small a individuals, and there will be homozygous recessive 160 which will be having an allele of small a individuals. Dear students, let's now discuss about the factors affecting gene frequency. Populations change over time. The number of individuals in population may increase or decrease depending upon food resources, climate, weather, and the availability of breeding areas, etc. At the genetic level, a population may change because of mutation, migration, selection, and random genetic drift. These natural processes alter allele frequencies, thereby changing the fundamental makeup of the population. So mutations produce new alleles, though the rate of mutation is too low to be noticeable. In case of deleterious mutation, selection counteracts the effect. Migration changes the frequencies because the immigrants from a population have different genetic makeup. So in a particular population, the gene frequency changes due to genotype change because of different immigration from other locality. The third one is selection reduces the fertility or survival of certain genotypes. In many populations, survival and reproductive ability are variable traits. Some individuals may die before they reproduce, whereas others may have many progeny. The differential contribution of progeny implies that alleles that are associated with superior fitness will increase in frequency in a population. The fourth one is that random genetic drift, that is chance difference, causes small deviations from the predicted frequencies, especially in small samples. These deviations, however, are expected and allowed for on the basis of statistical tests. Number 5 is the associative mating of either similar or dissimilar genotypes results in an excess of homozygotes and heterozygotes respectively, which change the gene frequency. Sixth one is the existence of subpopulations which are locally mating groups in a large population and may be due to some ethnic causes or class grouping as in humans or due to low mobility of some organisms in a large area. These phenomena increase the frequency of homozygotes, which is the result of inbreeding. Dear students, let's now discuss about the genotypic frequency. Genotype is the genetic expression of a particular trait or character and involves two or more alleles together to give rise to the particular expression. The genotype can be homozygous, that is alleles are of the same form like capital P, or heterozygous, that is alleles are of different forms, that is capital P, small p, and the genotype frequency measurement refers to how many times a particular genotype is expressed in a population at a given period of time. Thereby, the genetic relationship within a population can be determined. Genotype frequency is the frequency of the three possible genotypes which can occur in a particular population. They are the homozygous dominant, homozygous recessive, and the heterozygous genotypes. Each frequency of the genotype can be calculated by dividing the number of individuals that show a particular genotype by the total number of individuals in the population. In a randomly mating Mendelian population, the gene and genotype frequencies reach to equilibrium in a single generation. The Hardy-Weinberg law states that the gene and genotype frequencies in a Mendelian population remain constant generation of generation if there is no selection, mutation, migration, or genetic drift, but that hardly occurs in real world. So, homozygotes like capital A and small a that is recessive are produced by the union of gametes carrying similar alleles. The frequency with which a male gamete carrying allele A, that is capital A, has a frequency P, fuses with a female gamete carrying another allele with capital A also with frequency P will be P multiplied by P is equal to P square. Similarly, the frequency with which a male gamete carrying allele small a frequency Q fuses with a female gamete also carrying small a will be Q multiplied by Q is equal to Q square. Heterozygotes with capital A and small a are produced by the fusion of gametes carrying different alleles. The frequency with which a male gamete carrying allele A fuses with a female gamete small a will be P multiplied by Q is equal to PQ. Similarly, opposite way, the frequency with which a female gamete carrying allele capital A fuses with a male gamete carrying small a will be P multiplied by Q is equal to PQ. So, the total frequency of heterozygotes will be 2PQ. Dear students, let us now talk about the similarities between gene frequency and genotype frequency. Gene frequency and genotype frequency are measured within a particular population, preferably in a micropopulation. Both percentages are measured within a particular time period. Both values are expressed as a percentage. Both measurements are used to determine the genetic relationships within a selected population. Genotype frequency and allele frequency are two types of parameters that describe the genetic variation of a population in the Hardy-Weinberg equation. The sum of all possible frequencies is equal to 1. Dear students, let us now discuss about the differences between genotype frequency and allele frequency. The main difference between genotype frequency and allele frequency is that genotype frequency is the frequency of the possible three genotypes in a population. Homozygous dominant, that is capital A, homozygous recessive, that is small a, and heterozygous, that is capital A, small a, whereas the allele frequency is the frequency of the two types of alleles in a population. Dominant allele A and recessive allele small a alleles. Furthermore, the P square, Q square and 2PQ values represent the three possible genotypes, while P and Q represent two alleles in the population respectively. Genotype frequency refers to the number of individuals with a given genotype divided by the total number of individuals in the population, while allele frequency refers to the frequency of occurrence or proportions of different alleles of a particular gene in a given population. Thus, this is the main difference between genotype frequency and allele frequency. In Hardy-Weinberg equation, the genotype frequencies are P square, Q square and 2PQ values, while allele frequencies are P and Q. Hence, the type of constituent frequencies is a major difference between genotype frequency and allele frequency. However, both genotype frequency and allele frequency are important for the calculation of genetic variation of a population according to Hardy-Weinberg equation. In brief, the main difference between genotype frequency and allele frequency is the type of constituent frequencies. Dear students, let's discuss about the genetic inheritance and evolution. Genetic inheritance or heredity is the transfer of genes from parents to their offsprings. Evolution is the process in charge of genetically changing species, making them more adaptable or adapted to their environment, which ensures the survival of the species which are better fit to that particular environment. Natural selection is the process in which organisms that are more fit or best adapted and able to pass on their DNA through reproduction survive, and organisms that are less fit do not. According to natural selection, the organisms with favorable traits will succeed. Advantageous traits in the wild differ based on the species. For example, a male peacock seeks out female peahens during mating season. The mating ritual consists of a peacock's display of their feathers, often being pushed out and up to make them seem as large as possible, wherein they shake them in a sort of dance. Scientists have found this show helps peahens find their mates in the wild. Therefore, if peacocks possess large feathers, they are more likely to find a mate. Favorable traits are more likely to be passed to offsprings because mates of a species find these fit qualities appealing. This simple fact ensures that auspicious traits will be passed on to their offsprings by mating with partners who have these qualities. Allele frequency shows what traits are most favorable within a species group. Male peacocks that have tall and vibrant feathers tend to be more fit. Therefore, the frequency of the gene and alleles for tall and vibrant feathers will be high in a population because this trait is more likely to be passed on. Dear students, let's now talk about the genetic inheritance. Genetic inheritance is the process in which DNA is passed on from parents to their offsprings. All genetic traits are made up of gene. Each gene has two alleles, one from each parent, that is one from mother and one from father. A parent's particular genotype determines whether certain traits are expressed in offspring or not. Alleles can be either dominant or recessive, and dominant alleles have the ability to override recessive traits. If one parent passes a recessive allele and the other parent passes a dominant allele, both will be inherited. However, the likelihood of the recessive trait being expressed is minimal. Population genetics studies the frequency of alleles within an overall gene pool or a specific population of organisms. This subfield of genetics specifically looks at whether genes are changing the relationship of organisms and the factors that might influence change in the population. Dear students, let's talk about the allele frequency and evolution. Evolution is the genetic change within a group of species that happen over generations using various processes such as natural selection. Allele frequency is the visualization and evidence that evolution is taking place. By calculating allele frequency throughout the several generations, it is possible to see how groups of species change over time. Microevolution is the rapid form of evolution that occurs in a short amount of time. Sometimes these changes can be seen from one generation to next generation. Dear students, let's now discuss how the evolution occurs. The evolution occurs by mutation, that is the change in the DNA. Second, by migration, that's an influx of new individuals to the population. third, natural selection, that is, most fit organisms survive to pass on their DNA through offsprings. Fourth one is the genetic drift, that is, random chance an allele is not passed to offspring. The synthetic theory of evolution is the study of evolution in relation to gene frequency, which produces new species. This theorem analyzes change in a species population that happens via the evolutionary processes and proposes that however evolution happens, that variation will occur. Once variation occurs, the fittest organism will then thrive or survive. The term species refers to a group of organisms that are biologically and behaviorally similar and can produce offsprings. The other one is speciation. Speciation is when organisms within this group separate and create their own species. The four types of speciation are allopatric speciation, peripatric speciation, parapatric speciation, and sympatric speciation. Dear students, let's now conclude the lecture. The gene pool, which consists of the total number of genes in a particular population, constantly evolves as species undergo adaptations for environmental and other physical factors surrounding them. Hence, geneticists use change in genes and genotypes to study the evolutionary patterns over a period of time. The gene frequency and genotypic frequency are measurements conventionally determined through Mendel's theories, but advanced with the theories put forward by Darwin on evolution. Allele or gene frequency is a measure of the relative frequency of an allele on a genetic locus in a population. Genotypic frequency is the proportion of a particular genotype amongst all the individuals in a population. This is the difference between gene frequency and genotypic frequency. Dear students, it was all about today's lecture regarding genotypic and allelic frequencies. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.